Hello everyone, today we are going to talk about your tryptophan operon or TRP operon, which is basically concerned with the synthesis of tryptophan. That is an amino acid which is involved in the biosynthesis of proteins. So this is the structure, as you can see, the structure of an tryptophan amino acid in the slide. So yeah, in 1961, Jacob and Monod for the first time gave the concept of transcriptionally regulated system, which is called as operon. So this was first identified as a mode of gene expression controlled by the operons. Now, a polycystonic structural gene regulated by a common promoter and regulatory gene is referred to as operon. We have a lot of examples like TRP, operon, the lag operon. So basically here we are concerned with TRP operon. Before we move to the next slide, you need to know what is a polycystonic structural gene. So a polycystonic structural gene is basically an mRNA that encodes for more than one polypeptide. So let us see the structure of the operon. So it is clearly demarcated in the slide. It consists of promoter region, then we have operator region, and a structural genes or structural genes. Now besides this, we have a regulatory gene. It is not a part of operon, please keep that in mind. However, this regulatory gene is constitutively expressed. So an operon consists of three basic DNA components. One is your promoter, the other is your operator, and then we have structural genes. Now promoter is basically a nucleotide sequence which is being recognized by the RNA polymerase. So basically RNA polymerase recognizes the promoter region and binds to it to initiate the process of transcription. Now operator gene, as you can see over here, it's a segment between the promoter and the structural genes. Then we have the structural gene. The region consists of genes that are co-regulated by the operon. So a TRP operon or tryptophan operon is responsible for the synthesis of amino acid tryptophan when it is not available in the environment. Please keep that in mind. This is functional when in the environment your tryptophan is not available. So when tryptophan is not available, then only the TRP operon will be switched on. The TRP operon is made up of a promoter with an operator and 5 structural genes that encodes enzyme for the tryptophan synthesis. Now the tryptophan operon is regulated by the regulatory gene TRPR gene that is located at a distance from the TRP operon. Please keep that in mind that the regulatory gene is constitutively expressed or expressed most of the time. Okay, let us see the diagram and try to understand. This is the operon. In the first case, we will be talking about when tryptophan is absent in the medium. Now, this TRPR, a regulatory gene, as you can see over here, and we have talked about that it is constitutively expressed, so it undergoes transcription to produce an mRNA or messenger RNA. Then this messenger RNA will undergo translation to produce a protein, which is an inactive repressor. Please keep that in mind. It is an inactive repressor, and since it is inactive, so it cannot go and bind to the promoter region, and since it cannot go and bind to the promoter region, so what happens, the RNA polymerase will come and bind to it, to the promoter region, and this would result in the expression of the structural genes. So basically when this inactive repressor cannot go and bind to the promoter region, so the promoter region is very much available or free for the RNA polymerase to come and bind and initiate the transcription of the structural gene. Here you can see there are 5 structural genes, E, D, C, B and A. So this will undergo your transcription, and then it will produce the required protein or the amino acid. Now let us see a different case where tryptophan is present in the medium. Now as such, this regulatory gene will undergo transcription and will produce a messenger RNA, and that messenger RNA will produce again the protein. Now this protein is, again, it will be an inactive repressor. Now what happens when the tryptophan is present in the medium? So this tryptophan will function as a co-repressor and will go and bind to the inactive repressor. So once it goes and binds to the inactive repressor or the protein, then what happens, there will be a conformational or configurational change in the protein molecule of the regulatory gene product. So once there is a particularly what happens when there is a change in the conformation, so what you can see over here, that co-repressor will go and bind to the promoter region. Now apparently this co-repressor happens to be a tryptophan, so this brings about a change in the configuration of the protein, and this protein will go and bind to the promoter region, and since it goes and binds to the promoter region, so the promoter region is not available for RNA polymerase to come and bind. Since RNA polymerase cannot come and bind to the promoter region, so there is no gene expression or the expression of the structural gene. So in that case, there is no transcription and the system is switched off, or the operon is switched off. So please keep that in mind. In absence of tryptophan, the operon is on, and in presence of tryptophan, the operon is off. So the TRP operon is an example of repressible operon. It is on unless turned off by a repressor protein. So obviously until and unless it is turned on by a repressor protein, it is not getting repressed. Now the repressor protein is synthesized by TRPR. While the repressor protein is always present in the cell, it is synthesized in an inactive form. Now when a co-repressor is present, in this case tryptophan, it binds to the repressor protein in the allosteric site. So what are allosteric site? Basically these are sites where any molecule can come and bind. In this case it's basically the tryptophan which comes and binds to the repressor protein. Now when a co-repressor is present, in this case tryptophan, it binds to the repressor protein in an allosteric site. So this brings about a conformational change of the protein such that it can bind to the operator and block transcription by preventing the binding of RNA polymerase to the promoter. So in this way, the cell saves energy by not producing tryptophan when it is already present. So it makes sense, because when tryptophan is on or present in or very much available in the medium, why should the cell exercise energy to get its gene expressed and produce tryptophan when it is readily available? So this was all about your tryptophan operon. Thank you and have a nice time, and if you like my video, then please do subscribe.